Okay, this is standard 2.6, which is about mitosis. So we'll be looking at the cell cycle during this particular podcast, and then in the next one we'll be looking at the role of mitosis in formation of new cells. So you will be able to describe the 4 stages of the cell cycle and the 4 phases of mitosis and this process called cytokinesis. You'll be able to describe the structure of a chromosome. You'll recognize that mitosis produces cells that are identical to each other and to their parent cell, so you're making copies of cells during mitosis. And mitosis is used to either form new somatic cells or for asexual reproduction for certain types of unicellular eukaryotes. Okay, so we're going to be talking about in this podcast interphase, the sister chromatids and chromosomes, what a centromere is, and then the second one we will be talking about the stages of mitosis, prophase, metaphase, telophase, and anaphase, as well as some of the different structures that are involved during that process. All right, so we're going to start looking at the cell cycle. This is the stage of activity of all cells, so all cells go through a cycle basically of life. And we generally will define the cell cycle as being the beginning of one cell division to the beginning of the next cell division. So as the cell divides, goes through its whole life, and then begins to divide again, that is one full cell cycle. And different cells are going to spend different amounts of time at different stages within the cell cycle. So during the cell cycle, various things are going to happen. The major component of the cell cycle is the cell growing. So it's going to spend a good portion of its time just growing to the size that the cell needs to be. Once the cell has grown to its size and is doing all of its regular activities, it only can grow so big. So at that point, the cell will then need to prepare to divide, and then the actual division will happen. So the cell will grow, do its normal activities. Once it gets to a certain size, it then has to prepare itself to divide, and then it goes ahead and does that division, making 2 identical cells, and we call these cells daughter cells. That's just the word that they use when they're identifying the cells made by mitosis. Okay, so in the cell cycle, there are 4 stages that we're going to talk about, and this graphic kind of gives you an idea of what those 4 stages are. The first is called G1. During this stage, that is when the cell is growing and doing all of its normal activities. During S stands for synthesis. So that is the stage where we're going to actually be making copies of DNA. So we're doing DNA replication during that point. After that is G2. This is another growth stage. And during G2, we're actually going to be preparing the cell to undergo mitosis, which is the division. And then there is the M stage itself, which is comprised of both mitosis and cytokinesis, and that is when the entire cell is going to divide. So we're going to take a look at each of these stages in a little more detail. Okay, so we're going to start by looking at interphase. Interphase as a whole is the longest phase of the cell cycle. So this is where the cell is going to spend most of its life, and it is divided into the first 3 stages of the cell cycle that we just talked about. So G1, S, and G2 all comprise this stage of life called interphase. Now, some cells spend their entire life in G1, and they never leave G1 because they're incapable of dividing. Example of that would be your nerve cells. Once your nerve cells have been made, they're not going to make copies of themselves anymore. So some stay right in the G1 phase. Others go through this cell cycle fairly quickly, like once every 24 hours or so, like your skin cells. And they'll do the entire cycle. But most of the time for all of your cells, they are hanging out in interphase. And once they get to M stage, where the division happens, it usually happens pretty quickly. So let's take a look at what happens to G1. G1 stands for growth. And this is when the cell is going to perform all of its typical activities. So if it's a pancreatic cell, then it's making different enzymes, and it's making insulin, and it's making glucagon, and it's doing all the things that it needs to do, in its day-to-day activities. So we could be making different proteins. This is where all the different, when metabolism is going crazy, cellular respiration or photosynthesis, if you're talking about a plant cell, all of those regular activities that the cell is going to do are going to be happening during this G1 stage. And it is also at this point that the cell will grow up to its mature size. And that size of a cell is determined by how many other cells are around it and what the overall function is for the cell. But typically, once a cell becomes in contact with another one and starts to run out of room, that's when it has reached its mature size. and usually that is when it will get the signal that it needs to divide. So once the signal is sent that division needs to happen, we move into the S stage. S stage stands for synthesis, and this is where a copy of the cell's entire library of DNA is made, and this process is called DNA replication. So remember, DNA determines all of your traits. Everything that the cell can do is determined by its DNA. So if you're going to make a copy of the cell, then you have to make a copy of that DNA. So we make one big copy of all of the DNA during S stage. Okay, so if a cell does not duplicate its DNA before division, then it's not going to divide correctly. So this step must happen. DNA has to be copied before the cell can divide. Okay. The way it does this is it makes copies of its chromosomes. I should say of its chromosomes. So you're going to make all of your chromosomes. So if you think about a human, humans have 46 chromosomes in a regular cell. So if you were about to divide, if that cell was about to divide, you would double that number of chromosomes temporarily so that you would have 92 chromosomes. And then when that cell divides, you'd get 2 new cells with 46. So you'd have a doubling of your chromosomes temporarily. So let's talk a little bit, take a break here and talk a little bit about what your chromosomes are. So in your cells, during G1, when the cell is not dividing, it's not really doing anything fancy, your DNA is found in these really loose strands in the nucleus called chromatin. So basically it's kind of like a ball of string. So it's loose. It doesn't take a lot of energy for the DNA to be in this stage. It's kind of just a relaxed state. The DNA can easily make its proteins and do everything that it needs to do when it's in chromatin form. The problem with chromatin form is that if you were to try to divide the chromosomes that make up this chromatin, so it would be 46 strands of DNA, and you would try to split them up without making any mistakes and without them hitting each other, you wouldn't be able to do it. So before the cell can divide, you have to condense your chromatin down into chromosomes, and they become these individual little rod-shaped things. And so separating these chromosomes from each other would be a lot easier than trying to separate this whole mass and tangle all together. And that is why we don't make very many mistakes when cells divide, because they get into this chromosome form, and so when they separate, they can separate very cleanly. So taking a closer look at chromosomes basically are condensed chromatin. So you're going to take the chromatin and it's going to wind itself up and really make like a nice solid shape that is not all loose like the chromatin. You are able to see your chromosomes during prophase of mitosis, so that's the first stage of mitosis. We'll talk about that in the next podcast. And basically a chromosome is made up of 2 identical sister chromatids. So when a chromosome is going to make a copy of itself, so here you have one side of the chromosome over here, and when the chromosome is going to make a copy of itself, it makes an exact copy. And those 2 sides actually get attached to each other in the middle by a special protein called the centromere. So you have 2 sister chromatids, they're identical to each other, and you have a centromere in the middle. Now this is only when the DNA has been duplicated. So this is not what your chromosomes look like when you are not ready to divide. Okay, this is only during duplication. So the 2 sides of this chromosome are identical. In your regular cells, you would just have 1 chromosome. And again, it would be in that really loose chromatin form, that's the big ball of string, because it takes a lot less energy for the cells to be in this loose form than it does to be in this nicely compact form. All right, so once we have made a copy of all of our DNA, and we've had a complete set of DNA, two sets of it, we then can move into the G2 stage. And this stands for growth 2. And this is where the cell is going to prepare for division, so it's going to get itself ready to divide. And so a couple of things that happen. We're going to make copies of essential organelles, because remember, if you're going to make a new cell, it's got to have all the organelles that it needs to have. So the mitochondria will get copied. They'll make some copies of endoplasmic reticulum. They'll make sure there's enough ribosomes for the new cell. All of that stuff is going to get made in duplicate so that both cells have what they need. The cell will double in size typically at this point. Right before division, it'll get really, really big. And then also at this point, we're going to make a special organelle called a centrosome. And inside the centrosome is a centriole in an animal cell. So in plant cells, it's just a centrosome. In animal cells, it contains a centriole. Basically, this is an organelle that is needed for the process of mitosis to happen. So it has to be made at this point. It's not an organelle that we need during any other time. So it's typically not there unless the cell is going to divide. So during G2, we're going to make this special organelle so that when the cell divides, it can do its job. So this is what a cell would look like at the end of interphase. Right before the cell is going to divide, so we've got our centrosomes have been made, so they're going to be doing an important job for us. Our nucleus is full of a double amount of our chromosomes. Our chromatin's been doubled up. The nucleolus is still there. You have your nuclear envelope. So this is basically a cell that is ready to divide, getting ready to go, but it's not started, hasn't started dividing yet. Once it has gotten the signal that everything is okay and it's all ready to go, then we enter M stage. M stage stands for mitosis and cytokinesis. During mitosis, the cell's nucleus is going to divide into two identical nuclei. So mitosis is actually the division of the cell's nucleus. It is not division of the cell itself. So the nucleus makes a copy of itself. So if you think about it, this is where we're going to be taking all of those chromosomes in the form of our DNA, and we're going to split them apart. So those chromosomes that were in those X forms, we're now going to separate them into individual chromosomes. And they're going to each end up in their own cell in the end. Okay, and remember, in humans, we've got 46 sets of these. Followed by the division of the nucleus, then we are going to do the actual division of the cell, which is cytokinesis. Remember, cyto means cell. So we're going to divide the cell in two. So this is the cytoplasm is actually going to get divided, and we end up with two identical daughter cells because the mitosis portion has already happened, and they each have a complete set of the DNA. And then you have your two identical daughter cells. So the next podcast, we'll take a look at the specific things that happen during mitosis.